Okay, hello everybody and welcome to our ASPS, Australian Society of Plant Scientists Award ceremonies for 2020. This afternoon we're going to have 4 presentations, 20 minutes at each presentation with 5 minutes of questions. We've got technical support from Mary Williams, very grateful for that, run by the American Society of Plant Biologists. If you note at the bottom there, if you're having any technical troubles, please let us know in the chat box. When it comes time for questions for the speakers, please use the question and answer box in the Zoom there. At the moment, we're all muted and I will select questions and read them aloud as chair. Today we're presenting, Mary, if we could have the next slide. Yep. Okay, today we're having the presentation of the Jan Anderson Award. It's sponsored by the ARC Centre of Excellence for Translational Photosynthesis and CSIRO Agriculture and Food. The Goldacre Award medal is going to be awarded today as well, sponsored by Functional Plant Biology and CSIRO Publishing. The whole webinar is posted by Plantae, ASPMB. Very grateful for that. And I want to point out to you that IPMB 2021 has now been moved to IPMB 2022 to be held in Australia in Cairns from the 23rd to 27th of October, and we're hoping to welcome lots of people at that meeting. So keep checking it out. Okay, if we could go back to the previous slide, I'd like to introduce our speakers for today. I'm going to start with the Jan Anderson Award and give you a little bit of information about the Jan Anderson Award. Jan Anderson, this award is in memory of Jan Anderson, who was an esteemed woman plant scientist who unfortunately passed away in 2018. She was a role model to women in plant biology. Even as when I was a PhD student, I interacted with Jan. She was an amazing woman. She worked in the area of photosynthesis, on the structure of the photosynthetic apparatus and its heterogeneity. She found the heterogeneity in the chloroplast membrane. This award commemorates early to mid-career women plant scientists. It's an award that is accompanied by a $2,000 cash stipend and certificate, which are both in the post. This year there was a very high competitive, competitive group of applicants for this position, and it came to the point where the referees or reviewers for this award could not decide on who to give the award to, so we decided to award 2 joint winners this year, Dr. Sweetman and Dr. Kristine Crous. Crystal will give her talk first. Crystal did her PhD in 2010. She studied the area of grape metabolism, but recently her primary focus has been in primary plant metabolism and environmental stress, with a focus in the area of plant respiration. We now listen to Crystal's presentation for the award, and we'll follow with Kristine's, but I'll come back and tell you about Kristine just before she starts to talk. So, Mary, let's get going with Crystal's presentation.
Hi, thank you, Kathleen, and thank you to ASPS and the award assessors for selecting me for this award. It's really such an honour to be in the company of my co-award winner, Kristine, as well as past award winners, Monica and Eloise. Before I get started, I also want to thank the ASPB and Plantae webinar organisers for allowing me to pre-record this talk while I'm on maternity leave. And today I'm going to share some of my work from the last 6 years, focusing on the alternative pathway of respiration and how it could be employed to curtail abiotic stress in plants. So in order to support human life and growth on this planet, we need to find clever and new ways to improve plant productivity with increasingly limited resources. So the Green Revolution was highly effective at getting food in bellies, But the growth in yield is plateauing while population growth continues, and currently 1 in 9 people across the globe are undernourished. So we need to find more ways to sustainably improve crop yield on a finite area of land with finite water and nutrient supplies. And of course, climate change adds to the challenge because more difficult growing conditions and weather extremes also decrease crop productivity. And of course we want to minimise the devastating losses of life due to starvation and malnutrition across the world due to limited staple foods like cereals and legumes. But I thought I might also mention some of life's luxuries that will become harder to come by with the changing climate due to impacts on growth of, for example, coffea trees, grapevines, barley and hops plants, and cacao trees. So at the very least, climate change will affect terroir, so that is characteristics of a product that are defined by the growth location and its inherent environment. So for example, a classic big red Shiraz from the Barossa Valley here in South Australia may begin to lose its unique taste and aroma characteristics. So a little more on abiotic stresses and how they affect plants. So here we can see a summary of some different types of stresses that might be endured by plants, including water limitations and water logging, extremes of temperature, extremes of light, salinity in the soil, and all of these can lead to the formation of reactive oxygen species, or ROS. So ROS at low concentrations are quite beneficial. They can be used as signals to stress, allowing the plant to respond and adapt. However, overaccumulation of ROS during prolonged or extreme stress leads to a condition called oxidative stress, which results in cell damage and ultimately either programmed cell death or necrosis. And this is how we end up with a field of drought-stressed wheat looking like this. So processes that are involved in ROS formation or ROS mitigation can be important targets for trying to improve tolerance of plants to a whole range of abiotic stresses. So one such process is respiration through the electron transport chain in mitochondria, where ROS do overaccumulate during stress. So here is a schematic of the classical pathway of electron transport in mitochondria, which is common to essentially all eukaryotes, including humans, animals and plants. It consists of large protein complexes, 1, 2, 3 and 4, and some small mobile electron carriers, ubiquinone and cytochrome C. And then electron donors or electron carriers such as NADH donate electrons to these complexes, which each transfer electrons to each other, like a chain. And then at each step of the chain, these electrons transition to lower states of energy. The energy released is used by complexes 1, 3 and 4 to drive proton extrusion from the mitochondrial matrix across the inner membrane into the intermembrane space. And this leads to an electrochemical gradient that then drives ATP synthesis through the ATP synthase enzyme. And then ROS in the form of superoxide excuse me, are generated as a byproduct of the classical electron transport chain due to electron leakage at complexes 1 and 3, and also possibly complex 2 in plants. And then during stress, overaccumulation of ROS can occur due to over-reduction of this electron transport chain and can compromise the function of this whole pathway. In plants, there exist additional proteins in the inner mitochondrial membrane that can act as a bypass of the classical electron transport chain, and these are referred to as the alternative pathway of respiration, and they can act kind of like a safety valve to prevent overaccumulation of electrons within the chain, and thus help prevent overaccumulation of these ROS. So here is a very simplified version of the electron transport chain in plants. This time the classical complexes are shown in white, while the alternative pathway, or AP, is shown in pink. And this involves 2 different types of enzymes. So we have the alternative NADPH dehydrogenases that can bypass complex 1. These are located on either side of the inner mitochondrial membrane, with NDAs facing the matrix, so they oxidize matrix NADH and NADPH, and then NDBs, which face the intermembrane space and therefore oxidize NADH and NADPH from the intermembrane space, or essentially from the cytosol. We then have the alternative oxidase that bypasses complexes 3 and 4. So these enzymes do not contribute to the formation of the proton gradient and are therefore said to uncouple NADH oxidation from ATP synthesis, or also, in other words, they can be called an energy wasteful pathway. So there are numerous proposed roles of the AP in plants. I've listed some of them here. So first we have thermogenesis, and this is actually how the pathway was first identified, through heat production in the thermogenic arum lily, which assists pollination of the flowers. But there are also more complex roles proposed. For example, maintaining respiration if the classical complexes become compromised. Also bypassing sites of ROS generation to prevent overaccumulation of ROS during stress, and this can also protect the enzymes of the classical pathway. Also, there's a possible metabolic role in uncoupling NADH oxidation from ATP synthesis, which can enable continued operation of upstream respiratory pathways such as the TCA cycle that actually generate the NADH used by the electron transport chain. If we get too much ATP produced, then that causes a feedback inhibition on these upstream respiratory pathways. So the alternative pathway can also give us some kind of metabolic flexibility, if you like. So all of the proteins of the Arabidopsis alternative pathway have been identified. So there are 5 alternative oxidase isoforms and 7 alternative NADPH dehydrogenase isoforms. Of the alternative oxidases, or AOXs, the AOX1A protein is the of most interest and is the most studied because it is very stress responsive. An overexpression of AOX1A improves stress tolerance, while knockdown of AOX1A impairs stress tolerance. The alternative NADPH dehydrogenases, or NDs, are less studied, but of these 7 different isoforms, NDB2 is the most stress responsive, and it's also co-expressed with the very stress responsive AOX1A. Knockdown of another external facing NADPH dehydrogenase, NDB4, induced NDB2 and AOX1A transcription protein and actually improved stress tolerance. However, the effect of directly manipulating NDB2 expression in plants has not yet been done, and so the effect of altering NDB2 expression on plant stress tolerance is as yet unknown. So this was one of the main kind of drivers behind the work that I've been doing. I wanted to look at whether manipulating NDB2 protein levels can have an impact on the stress tolerance of Arabidopsis plants. And to do that, we generated a number of transgenic Arabidopsis lines. This first one that I'm going to talk about is just a TDNA knockout line that I obtained from the Salk Institute, where the NDB2 gene has been interrupted. So here we can see a Western blot where the NDB2 protein is essentially lost. And in this graph you can see both the transcript and protein are much lower in this NDB2 knockout line compared to the wild type. And this led to a very significant decrease in oxidation of external NADH in purified mitochondria. So what this means is that this NDB2 protein is really the main external NADH dehydrogenase in Arabidopsis mitochondria. So we also developed NDB2 overexpression lines. And so we can see here that levels of transcript and protein were higher in these overexpression lines compared to wild type, in some cases by as high as 150-fold, but with some variability between lines. However, in none of these overexpression lines did we see a translation to increased activity levels. So we have high levels of protein expression, but this is not resulting in higher levels of activity. And this was puzzling. Is the NDB2 protein inactive somehow? Is it being degraded? We weren't sure, so continuing on with this work, we developed some transgenic lines that were overexpressing both AOX1A and NDB2. So we introduced an NDB2 overexpression construct into a line that already was overexpressing AOX1A. So this blot shows the accumulation of AOX protein in the AOX1A overexpressor line. And then in our dual overexpression lines, we can see the higher level of AOX and NDB2 protein compared to the wild type. And we can see that in this graph here as well. So we've got higher levels of NDB2 protein and AOX1A protein, and this did give us increased activity rates of external NADH oxidation in our mitochondria. This was quite exciting. So for some reason, the overexpressed NDB2 protein would only be active if AOX1A protein was also overexpressed. And so this suggested that there might be some kind of link between these two proteins. And now I had a range of plant lines with altered NDB2 activities that I could use further for determining whether altered NDB2 would have any impact on a stress response. So I had a look at these lines in more detail. So first, and I don't have data for this to show you, but I had a look at growth of these lines under normal conditions. And I took a lot of measurements, but in general the consensus was that the single AOX1A and NDB2 overexpression lines had delayed development and some smaller root phenotypes, whereas the dual overexpression lines, where we overexpress both AOX1A and NDB2, these typically looked more similar to the wild type plants. And what that suggested was that an imbalance of NDB2 and AOX1A protein could cause some detrimental effects on the plant growth. However, I'm more interested in looking at these transgenics under stress experiments. So here is an experiment with the NDB2 knockout line. So this is shown here, and this was alongside an AOX1A knockout line that was also previously characterized. So in this experiment, I grew the plants for a few weeks, then withheld watering for a week, and then after that continued to withhold watering, but also turned up the light intensity to what we called moderate light. And the idea behind this stress, which we took from this previous paper here, is to expose the plants to a photoinhibitory condition. So specifically, drought stress will cause the plants to close their stomata, limiting water loss, but also limiting carbon dioxide transfer to photosynthetic cells. And then on top of that, the higher light intensity kind of becomes too excessive for the plant to use for photosynthesis, and this causes damage to the photosynthetic machinery. So this treatment was ongoing until the plants were extremely stressed, and then upon rewatering, the knockout lines could not recover. So that is, they had kind of passed some threshold or point of no return and succumbed to this stress, whereas the wild type could still recover. So these knockout lines had an impaired stress tolerance in this case. So I repeated this experiment with the overexpression lines, and this time I extended the treatment for a longer period of time until the wild type plants reached this point of no return. And excuse me, in this experiment, the single NDB2 overexpression lines behaved essentially as wild type, while the AOX1A overexpression line and the dual overexpression lines were still able to recover. So these lines were better able to withstand the extended photoinhibitory treatment and had not yet reached that point of no return. And I will remind everyone that these NDB2 overexpression lines, from previous slides, we saw that while there was increased protein, there was no increase in activity in these lines. So this may explain why there's no difference from the wild type. So the results from these experiments led me to some conclusions about the function of NDB2 in Arabidopsis. Firstly, we now know that NDB2 is the main external-facing NADH dehydrogenase of Arabidopsis mitochondria. It's responsible for most of the activity. Secondly, NDB2 seems to be functionally linked with AOX1A. So that is, NDB2 activity in vivo requires AOX activity. There may even be some direct link and a formation of a complete alternative pathway that bypasses all of these other complexes, especially during stress. So that's quite exciting. And then thirdly, NDB2 and AOX1A are both required for coping with photoinhibitory stress. However, tolerance could only be improved by overexpressing AOX rather than NDB2. So to describe how the alternative pathway of respiration may help mitigate photoinhibition during combined drought and moderate light stress, I've made this small animation. So it involves cooperation between chloroplastic and mitochondrial biochemistry and will involve complex signaling to minimize ROS production and cellular damage. So we can see here that first off, drought stress leads to lowered stomatal conductance and therefore a limitation of carbon dioxide in the chloroplast and therefore a limitation on dark reactions. And then on top of that, we blast them with a higher light intensity, which promotes the light reactions, leading to accumulation of reducing equivalents, ATP. And so because these reducing equivalents can't be then used in the dark reactions, they will accumulate and can lead to ROS overaccumulation. And so reducing equivalents from the chloroplast can be translocated to the mitochondria through various pathways, and so we can get now an accumulation of NADH in the mitochondria, leading to an upregulation of respiration and therefore increased ATP. However, ATP then provides a feedback inhibition on respiration, which can cause reduction of the electron transport chain and therefore overaccumulation of ROS. But what we have here is if we increase NDB2 and AOX1A protein and activities, we can kind of divert this NADH away from ATP production and therefore hopefully mitigate some of the overaccumulation of ROS. And so this is what I think happens in, at least in Arabidopsis, but now what I really want to do is look at this pathway in more important crops. And so recently I've been looking at legumes, specifically chickpea, and I will show you some of this work quickly. So chickpeas are highly nutritious. They can be grown sustainably, so in symbiosis with nitrogen-fixing rhizobacteria in the soil. So for this reason, they're quite interesting, and also they are sensitive to abiotic stresses. So growth and productivity could be improved. And after some bioinformatics work, I realised that chickpea, as well as 15 other species of legume that I screened, all have a different gene family structure of AOXs compared to all other groups of plants. That is, they only have one AOX1 isoform and multiple AOX2 isoforms. And here we have some graphs looking at AOX and ND transcript levels in 6 different chickpea cultivars grown in soil. The top graphs allow us to compare the relative level of expression between genes. So in the case of chickpea, a gene that we called AOX2A is the most highly expressed isoform. And this seems to be common between all legumes. There is a very highly expressed AOX2 isoform, especially in photosynthetic tissue. And what we can see here is that AOX1 is barely even visible on this chart. However, when we look down here, we can see in this graph, which shows transcript level as a function of fold change in salt stress plants compared to control plants, we can see that the AOX1 is actually the most stress responsive isoform. We can also see that NDB2 and NDB4, as we've called them, are the most stress responsive NDs of chickpea, but these are also expressed at a very low level relative to some of the other NDs. And this AOX2A transcript or protein is expressed so highly in leaf tissue that the stress responsive increase in other AOX transcripts is often actually not detectable at the protein or activity level. So the cultivars from the previous slide, I divided them into two groups, high and low sodium accumulators. Okay, so this was a salt stress experiment. And we can see a significant difference in the sodium to potassium ratio in our high sodium accumulators compared to our lower sodium accumulators. And the other thing that I saw was that the high accumulators also showed much more upregulation of alternative pathway genes compared to the low accumulators. So this suggests that there's some link between sodium accumulation or sodium transport and the alternative pathway in chickpeas. However, I could not find any relationship between alternative pathway transcript levels and various measures of salinity tolerance. What I did observe was a positive correlation between transcript levels of the AOX1 and the NDB2 during salinity stress. So this relationship was time dependent, and that's not surprising because upregulation of these AP transcripts in response to salinity was also transient and peaking for the most part at around 9 days after the initial salt addition. So this is interesting because this shows kind of a similarity to the relationship that's seen in Arabidopsis alternative pathway genes. So now I'll talk a little bit about where I plan to take this in the future. And because chickpeas have inherently high AOX capacity of this kind of energy wasteful pathway, there is some potential for energy saving by either inhibiting or knocking out the AOX2A gene, but leaving the stress responsive isoform. So this could actually lead to improved chickpea growth or even yield, ideally. Furthermore, I'd really like to link specific alternative pathway proteins with stress tolerance in legumes. So far we've linked the AP transcript increases to sodium accumulation. However, this was not an indicator of stress tolerance, so more work needs to be done here. And ideally we can find ways to use the alternative pathway to equip chickpeas and other legumes with a mechanism to more rapidly respond to abiotic stresses and therefore enhance their tolerance and improve productivity during difficult growth seasons that may have unusually warm or cool temperatures or unusually high or low rainfall, or for expanding growth into regions that have more saline soils. Circling back to the Arabidopsis work, we know now that the AOX1A and NDB2 proteins are important for facilitating stress tolerance under photoinhibitory conditions. But we don't know the mechanism for that, which is something that I want to explore further. And I'm also looking into some enzyme kinetics work and in vivo activities. So here are my references, and finally I'd just like to thank all the members of my lab and co-authors at Flinders University, the ASPS, for giving me this opportunity to present to you today, and I want to acknowledge the Legumes for Sustainable Agriculture Research Hub, which is funded by the ARC and GRDC, and this is where we get our funding to do our work with the chickpeas. And thank you.
Okay, thanks Crystal, and could I just remind anyone if they want to ask Crystal a question to pop it into the queue, type it into the Q&A list. Crystal, thank you very much for that very interesting presentation. I do have a question for you that's been put into the question and answer box. It's coming from Vanessa Molino. So I think we've got time for just a couple of quick questions. Vanessa's asking, could NDB2, thanks for the interesting talk, could NDB2, how do you think they are actually linked? And do you think there's a possibility that NDB2 could be a mobile protein that just associates with AOX during stress?
Thanks, Vanessa. Sorry for the noisy child. That is a great question. And as yet we don't know, but there have been some studies that use native page to look at complexomes in plant mitochondria. And there seem to be some potential physical associations between some of the alternative pathway proteins. So it is very possible. I think at this stage, none of them have specifically noted a physical association between AOX1A and NDB2, but we would be very interested to see if that actually does occur. Yeah, so that's a great question.
Okay. I forgot to mention that Crystal's actually been quite sick and recorded her presentation, and she's also the mother of a new baby, only a few weeks old at the moment. So well done, Crystal, for dealing with all that and recording your presentation.
Sorry for all the coughing during my talk.
It's okay, we know you don't have COVID. I've got a question here from John Evans. Is there any evidence that the alternative pathway is wasteful under unstressed conditions?
That's also a good question. Actually, I'm not sure. So one of the things about the alternative pathway that I guess is kind of useful in this regard is that it's subject to post-translational modification and activation. So it seems that, well, possibly a lot of the time, the alternative oxidase at least may not actually be active in vivo unless there is maybe over reduction of the electron transport chain, or there is kind of a need for it to be active. And that occurs in particular through organic acids, mostly pyruvate. So yeah, so there is kind of this extra level of control that may prevent it from being energy wasteful in the absence of stress. But yeah, that's another good question.
Crystal, there's another question from Monica Doblin. What happens to NDB2 or the double overexpressing lines when electron transport is disrupted? Sorry,
I'm not sure. I'm not sure I know what that.
So when the electron transport chain is disrupted, do they cope better? I guess maybe Monica might mean.
With some inhibitors, or?
Yeah, maybe.
I'm not sure. Sorry.
That's okay. That's okay. Crystal, there's a, oh yeah, that's what she meant, biochemical inhibition, she's written. Yeah.
Yeah. I haven't actually looked at this yet. So I can say that in vitro doing activity assays with the mitochondria from these dual overexpression lines, that if we inhibit the alternative oxidase and then try and measure the NDB2 activity, then we do see a reduction in the NDB2 activity. Yeah, so I guess that doesn't go all the way to answering your question, but it's something that definitely we should be looking at.
Yeah, or like cyanide, yeah. Crystal, there's a few questions in the chat box, but I think we need to be moving on. Could you have a look in the chat box and perhaps reply to these questions? They're quite interesting questions about breeding. And I'm sorry, Eloise and Peter, that we don't have time to cover them. So
yeah, I will do that. Yeah, thank you.
Yeah, do that, because then we can all see the answers. That'd be great. Okay.
All right.
That's
Thank
good.
you.
Thanks, Crystal. Thank you. And I will applaud. Now, the next speaker, Kristine Crous, our joint award winner for the Jan Anderson Award. She gained her PhD in the University of Michigan, has worked at ANU and University of Western Sydney, and currently there as a DECRA fellowship. Kristine's area is in the plant responses to atmospheric CO2 and looking at the implications of global warming within that too. So I would like to now welcome our next speaker, Kristine Crous, to give her presentation for the Jan Anderson Award. Thank you, Kristine.
Thank you, everyone. I'm also really honored to be able to receive the Jan Anderson Award. And yeah, I've been at University of Western Sydney, and I've been measuring temperature responses of photosynthesis and respiration in rainforest species as part of my DECRA award. And this is because I've always been interested in plant responses to climate warming. And I've done that for quite some time now in eucalyptus species and recently in rainforest trees. And so this talk, I will run you through how rainforest species are coping in the field under current temperature conditions. Then I will discuss how they respond to warming. And then at the end of my talk, I will briefly talk how plastic some of these rainforest species are. So in Australia, we're really lucky because we don't only have tropical rainforests, but we also have subtropical, warm temperate, and cold temperate rainforests, all of which come with their own set of species. And most of these species can actually be related back to the Gondwanan supercontinent. So these species are really ancient, and they were once extensive across the globe and also in Australia. And so basically these lineages have been around for over 140 million years. And that means that these rainforest species have at least seen some climate change in the past. However, in the last 10,000 years or so, rainforests have been enjoying stable temperatures year-round, especially in the tropics. And so the question really is, despite the past, how will rainforests cope with future climate warming? And if they don't cope, what does that mean in terms of species loss and global carbon uptake? So are rainforests at risk with warming, especially because we know that perhaps the warming in the tropics might not be as severe as the warming at higher latitudes? And the thinking is that tropical rainforests are adapted to thermally stable conditions, and therefore they may have less capacity to adjust to warmer temperatures compared to temperate rainforests, where we have more seasonal variation. We also know that rainforests are playing an important role in the global carbon cycle because these forests soak up CO2 from the atmosphere. And if we don't know how they're going to respond to warming, then that creates uncertainties in future carbon uptake predictions. So modelers have certainly pointed at the great uncertainties regarding the temperature responses of photosynthesis and respiration. And that's especially so in rainforest trees, mostly because of the scarce data. And this knowledge gap prevents us from understanding how rainforests will adjust to climate warming and by how much, and whether this adjustment is different in temperate rainforest species versus tropical rainforest species. A little bit more on temperature response curves. So you can measure the temperature response of photosynthesis, or you can look at the underlying processes of photosynthesis, meaning the maximum carboxylation rate or the maximum electron transport rate, and look at their temperature response. So for photosynthesis, you can measure this at different temperatures. That gives you a curve with an optimum. So where the photosynthesis is at its maximum, the temperature associated with that is called the temperature optimum of photosynthesis. And biochemical theory suggests that we expect increased photosynthetic capacity with warmer temperatures. And that's because at higher temperatures, enzymes are running faster, so we expect higher rates. We also expect an upward adjustment of the temperature optimum with warmer temperatures. And that assumes that plants are thermally adapted to where they grow. So what do we actually expect for warming responses? So on the left here, I have shown again the temperature response curve of photosynthesis, which has a clear optimum. And I also show the temperature response curve of respiration, which is very temperature sensitive because it has an exponential rise. The plants are currently operating around the temperature optimum here, which is indicated with the blue triangles. And so at a warmer future temperature, they could operate beyond their temperature optimum. And therefore, that means a reduction in photosynthesis or reduced carbon uptake. At the same time, they increase their respiration, so they're also losing more carbon back to the atmosphere via respiration. And that then means less carbon for plant growth. Now, plants don't really like that they don't get as much carbon for growth, so they tend to adjust over time to maintain that carbon gain. And so in photosynthesis, that happens by a shift in temperature optimum, as you can see here, towards higher temperatures. And for respiration, we change the temperature sensitivity. And that means really that the slope of this exponential relationship is much reduced, is much shallower. And that then means that the respiration rates are much lower at this warmer temperature compared to what they would have been if this adjustment had not occurred. So now we're going to look at how rainforest species cope in the field under their current temperature conditions. So I measured 13 rainforest species in 3 different sites. I measured 3 cold-temperate species in southern New South Wales near Batemans Bay, a mixture of cool and warm temperate rainforest species in northern New South Wales, south of Brisbane. And then I have measured some tropical species in the Daintree north of Cairns. And my choice for these sites are specifically chosen at the warmer edge of the distribution, because if there would be any temperature stress in current conditions where these species are occurring, you would expect it to see it at the warmest edge of their native distribution. And then we also all know that rainforests tend to occur in their own microclimates. So I've also measured temperature and humidity conditions, and basically that's what we're seeing in the next slide. So we have air temperature on the y-axis and date on the x-axis, but the dates don't line up. So to guide your eye, I have created or highlighted the summer months in these light gray boxes for each of these sites. And the reason why the x-axis doesn't line up exactly is because they were measured over slightly different timelines. So in tropical Queensland, we have an average summer temperature of 27 degrees and a maximum of 34. And the northern and southern New South Wales sites were actually quite similar, with an average summer temperature of about 20 degrees C and a maximum summer temperature between 30 and 32. And here are all the temperature response curves of photosynthesis that I've measured for these 13 species. And you can see the temperature optima indicated by the red arrows. So in the tropics, we find 2 temperature optimums, one at 33 degrees C, and that's for these 2 trees that stick way out of the canopy, and they're dominant trees, compared to species that are just emerging out of the canopy they have a temperature optimum of 30 degrees C. In North New South Wales, we also have 2 temperature optima, and that's because the warm temperate species have a higher temperature optima of 29 degrees C, whereas the cool temperate species have a temperature optimum of 25 degrees C. And this was for the red and yellow lines here, which are Notophagus moriae and Quintinia zebrae. However, this 25 degrees C temperature optimum for cool temperate species is also the same for the southern New South Wales side that you can see here. And now if we overlay the information from the previous slide with the average and maximum summer temperatures, we can see that rainforest species are well adapted to the current climate because their temperature optimums are laying mostly within the average to maximum summer temperature range. Except for one species here, Tasmania lanceolata, who has a temperature optimum of 17 degrees C and basically is operating beyond its temperature optimum during the summer. You can also look at this in a slightly different way, so via probability density functions. And so basically here on the left, I have repeated the information of the temperatures that I showed on the previous slide. And so you can basically now see that if you use a temperature optimum of 30 degrees C in the tropics, that only less than 1% of the time does the air temperature exceeds that temperature optimum of 30 degrees C. This is also the case for northern New South Wales. If you take a temperature optimum of 25 degrees C, then less than 1% of the time is that temperature optimum exceeded. It's a little bit higher in the southern New South Wales side, where we see 2.4% of the time exceeding 25 degrees C. But if your temperature optimum is 17 degrees C, then you're exceeding the temperature optimum 30% of the time. So how do rainforest species cope in the field? Well, temperate and tropical rainforest species are both well adapted to the sites where they occur, even at the warm edge of their distribution. Now, rainforest plants operate near their average growth temperatures, and they have some buffer to cope with warmer temperatures. However, there's one species, Tasmania lanceolata, who seems to be operating beyond its temperature optimum in the summer. Now, we can expect that if these species are well adapted to the places where they occur, then we would expect to see an increase in temperature optimum with growth temperatures. And that's exactly what we see here. So we see a positive relationship between the temperature optimum of photosynthesis on the y-axis and the growth temperatures on the x-axis. Except for that one species, Tasmania lanceolata, who seems to have fallen off the relationship. And then here we also see the relationship between temperature optimum photosynthesis and temperature optimum of electron transport, which is one of the 2 underlying processes of photosynthesis. So you can clearly see that those processes influence one another. And the electron transport is the most temperature sensitive process of the 2. So we see now that we see the same trend happening across the globe. So in a recent study of Kumaratonge et al., they also found a positive relationship between the temperature optimum of photosynthesis and the temperature optimum of growth, with low temperature optimums in the Arctic tundra, all the way to higher temperature optima in the tropical rainforest species here in orange, with the higher growth temperatures. So basically this shows that how species are coping in the field, but it doesn't tell us how they respond to warming. So how do rainforest species cope with warming? In order to answer that question, we set up an experiment with 2 temperate, 2 subtropical, and 2 tropical rainforest species, and they were grown at 4 growth temperatures each. And you can clearly see that we see again that positive relationship between temperature optimum of photosynthesis and growth temperatures. And this was especially so in tropical and subtropical species indicated in the red here compared to the temperate species. And it's in part because these two were also exposed to higher temperatures. However, what we didn't expect to see was a very strong reduction in photosynthetic capacity. Again, the maximum electron transport rate indicated as Jmax. And so for tropical and subtropical trees here in the red, you can see a very strong reduction with warming in Jmax. Whereas there was not that much change with warming in temperate trees. Now this reduced electron transport rate related to reduced photosynthesis rates and ultimately related into smaller plants and smaller leaves, especially in these really high growth temperatures. Now, of course, if you have a range of growth temperatures, you can also evaluate the magnitude of warming. So here I have done that by calculating the percent difference in A net compared to their home temperatures and the percent difference in electron transport rate. And in both of these, you basically see the same trends where most species seem to cope okay with 3.5 degrees C warming. But then if you look at 7 degrees C warming, you start to see different trends where temperate and subtropical species seem to be handling that okay, but there's a strong reduction in tropical rainforest species for photosynthesis. And that is mirrored in the electron transport rate, where at 7 degrees C, temperate and tropical species are doing quite well, but there's a strong reduction in electron transport rate in the tropical species. And then I've talked about photosynthesis so far, but we've also did some leaf respiration in this experiment. We're looking at R25, which is respiration measured at a standard temperature of 25 degrees C. And we looked at the temperature sensitivity Q10. And here we could see that tropical species reduce their rates at 25, but not temperate species. So the tropical species are indicated in this red oval. And if you look at the similar symbols, then you can see that each of these symbols declined with warming. Temperate species had also higher temperature sensitivity. So tropical species showed 24% reduced reduction in temperature sensitivity in tropical compared to temperate species. And that relates basically to this concept here, where the tropical species has reduced their temperate sensitivity compared to temperate species. So in summary of the warming experiment, we found reduced photosynthetic capacity with warming, which was particularly expressed in Jmax in this experiment. We found reduced respiration rates in tropical species, likely to curb carbon loss. Tropical species can likely cope some with 3 degrees C warming, but not much beyond that, while temperate species have capacity to cope with larger temperature increases. We found an increase in temperature optimum of photosynthesis with warming, but this came with reduced photosynthesis rates, especially in tropical species. So now the last part of my talk will show you how plastic rainforest species are in the field. And basically I try to use spatial gradients to look at warming responses. And I used 2 species here as an example. So one is the Antarctic beech, Nodophagus moriae, with its native distribution shown here. And the other is coachwood, Ceropetalum apetalum, with its native distribution here, taken from the Atlas of Living Australia website. And basically I measured the same species along its native distribution in these 3 locations. And so what did we find out? We could see that Nodophagus moriae showed the same temperature response no matter where you measured it. So Barrington Tops is at its southern native range, while the border ranges are at the most northern native range, and Mount Annan Botanical Gardens is outside its native range. And so no matter where you measure that, you see the same temperature response curve with overlapping confidence intervals. So the 95% confidence intervals are indicated by the dashed lines. For Coachwood, it's a different story, where we clearly see a difference with higher rates in the Blue Mountains compared to lower rates in the northern ranges, in the northern side where it's in Nightcap National Park. And so basically, in conclusion for my talk, rainforest trees are well adapted to their current climates. Tropical rainforest species can cope some with moderate warming, probably around 3 degrees C. Temperate rainforest species can cope with a broader range of warming temperatures than tropical trees. That was clear from our experiment. Notophagus is not adjusting to higher growth temperatures. And Tasmania lanceolata in New South Wales is operating beyond its thermal limits. And with that, I want to thank my PhD students, Zineb Shuri, for helping with the warming experiment on rainforest species. And I also want to thank my mentors to help me become the scientist I am today, and for inspiring me to work on plant responses with climate warming. And of course, I also acknowledge the ARC and Western Sydney University for support. And with that, I'm happy to take some questions.
Okay, thank you, Kristine. Thanks for that really good talk. I'll give you a clap too. Very, very interesting, sort of a little bit scary, isn't it, with what's coming on in the future. Do we have any questions from our attendees?
Well, there's some coming in, but there seems to be on the chat.
Yes, here we are from Eloise. If temperate species can adapt to higher temperatures, is fire the biggest risk?
Yes, I mean, there's always a risk for fire. We've definitely seen that last year. So obviously under extreme conditions, we're gonna see more fire risk. And if they get into the rainforest species, then they will be slower to adapt. However, that being said, not all is lost, because rainforest species are also good at resprouting, and therefore they are able to recover some from the bushfires. And I guess that's being measured right now as we speak in response to the previous year's bushfires.
Okay, so there's a question from Ross in the Q&A panel. Sorry, I haven't seen, here we are. It's from Ross Gledo. Is the tree that was different in photosynthetic response in the field study, is that more common further south? Could this New South Wales occurrence be already an outlier? Right, do you follow that, Kristine? Do you want me to read it again?
Yes, please. I'm wondering.
The tree that was different in the photosynthetic response in the field study, is that more common further south? And could this New South Wales occurrence already be an outlier?
Yeah, so I definitely think so. We're talking here about Tasmania, who had a very low temperature optimum. And so I think yes, it's already an outlier in New South Wales. Otherwise, we wouldn't see the fact that it's not so well adapted, and that it does is operating beyond its temperature optimum in summer. So I don't think that that's going to be more common south, because as you go more south, then you come more into its native range of Tasmania lanceolata. So I would expect that if I were to measure that species further south, that it would be better adapted to the cooler climate conditions there. But it would be great to see and measure what its temperature optimum is there and how much it changes throughout the range.
Good, good, thanks. Another question from Peter Ryan. Can you separate temperature and humidity effects?
Not so much in the field. That's a well-known problem, but basically what you do during these temperature response curves is you try to keep the humidity as moist as possible, which basically for any temperatures up to 35, I was basically able to keep the same humidity, but then once you go over like to 40 degrees C, then you could see that the chamber was getting drier, but I managed to keep it around, 45 to 50 degrees percent humidity. So I think that yeah, I can't separate it because it's in the field, but I think that I try to minimize it.
Okay, thank you. Thank you. So I think that we have no further questions. So Kristine, thank you so much for a really interesting talk. Thank you. And clap again.
Glad that you enjoyed it.
Well, certainly did. So well done, ladies, for two great presentations. We're now moving on to our next award. It's the Peter Goldacre Award. Our winner this year is Alex Wu. The Peter Goldacre Award recognises an early career researcher 3 years, past 3 years and within a limit of 10 years after their PhD. It's been established in memory of the scientist Peter Goldacre. He was a young scientist, a founding member of this society. He passed away at an early age of 34, unfortunately due to cancer, and this award commemorates his memory and keeps our thoughts of this going. It's sponsored, as I say, by Functional Plant Biology, CSIRO Publishing. It comes with a cash award of $2,000 and a medal, and if we were at the conference, I'd be handing this to Alex Wu. Alex has a double bonus because he was last year's Functional Plant Biology Best Paper Award winner as well, and he missed out on giving his presentation that year, so we're having a double bonus from Alex this year. Alex completed his PhD in 2015 from the University of Queensland, and he's interested in the area. He now works in the ARC Centre of Excellence for Translational Photosynthesis, and he's interested in modelling the processes that are involved in optimising plant growth. And Alex, I invite you to give us your presentation for your award.
So it's really my great honor to be receiving the Peter Goldacre Award, and thank the ASPS and the committee for recognizing my work and giving me the opportunity to present today. And today I'll be talking about crop modeling. And it's been a real privilege to be working with top scientists in the field in putting together this modeling work. So a bit about myself, how did I get into modeling? And I've always been fascinated about making predictions. And in my postgraduate study, I stumbled on mathematical modeling as a best way to make predictions. And the way to do that, you would build a mathematical model using your understanding about the system, and you make predictions about your observations, which you compare with observations, and then you refine your theory. And this has been a research mantra throughout my research career. Here on the left, it was my work in starch modeling, where I've considered a few the enzymes that makes the starch molecules, and connecting that through mathematical models to predict its structure. And this similar research theme translated to my current postdoc role in looking at crop models. And this is the focus of my today's talk. So just a quick overview slide on where this talk is going. So we all agree that there's going to be a significant shortfall of global food production in the near future. And in order to solve this issue, there's a lot of vibrant research going on in plant science and crop sciences. And having a capability to predict is actually a key to accelerate crop productivity improvement. And I'll be talking about the basis for doing these predictions. And also in the game recently is genetic engineering. And it presents new opportunities for improving crops. But besides the challenges in doing the actual engineering, there are also other equally important challenges related to genetic by environment by management complexities, which I will talk a bit about today. And in order to really understand this complexity of the system, we need to develop dynamic models that reliably connect processes across scales. And that's the tool that I'll be talking about, which is the cross-scale modeling. And hopefully show you guys that by doing this sort of modeling, we can enhance our understanding on the G by E by N complexities and use the model to explore new opportunities for crop improvement. And in the end, I'll make some predictions about photosynthesis. So can we predict crop grain yield, and what's the basis for prediction? Well, there's been a large body of work on crop modeling. I think even before I was born. And Graham Hammer, the best of the best in the field, has been doing crop modeling for a long time. And it's my privilege to be able to work with him over the past 6 years on sharpening my skills on crop modeling. So what crop model does is, well, it's basically a collection of a lot of equations, and I don't need to show you more, but you get the idea that all the processes that's happening, how the crop is interacting with the environment, with the management, and its underlying physiological determinants. We can capture that through functions that captures the responses of these interactions. And by having this dynamic model, we can predict how the trajectory of the attributes of your crop would go over a growing season. And here's a simulation of a sorghum crop over a season. We can predict the total biomass, the grain here, and then the stem mass and leaf area. So what this allows us is helping us to really understand the process that's underlying crop yield. And by comparing our simulation with the observation, we are ever so more confident about our understanding of the system. And one important applications of this is we can harness these understandings to unravel the complex G by E by M interactions and make evaluations on different traits. So the reason we're all doing this is there are concerns about food security and it's really driving plant and crop sciences. And genetic engineering is also in the game. It provides us with new opportunities, but however, there's little promise of this genetic engineering being realized for key complex traits. And because it's complex traits, it involves a lot of underlying mechanisms. And trying to understand the yield responses for a cultivar that is currently available, it's a very challenging job. And adding on to that, it is just unknown to know, well, what's the yield response of certain changes in photosynthesis in different environments. And I picked photosynthesis here, but this challenge really, it applies to every other yield determining processes. And the reason, as I said, we don't fully understand the G by E by M interactions, and our best knowledge at the moment beyond the biochemical pathways or the biochemical pathway levels remains largely empirical. And this is not to say that enhancing photosynthesis is a waste of time. Of course, it's not, because when we break grain yield down into its components, resource conversion is the one dragging behind, and resource conversion relates to the complex biochemical and leaf level processes in photosynthesis. And there are pretty good evidence on why higher photosynthesis gives you more yield. But we just need to better enhance our understanding of where this can take us. And so what we really have to do is linking photosynthesis with crop yield and having all these interactions working in the background. And this would be a way to address the complex G/E/M interactions that I've been talking about, and hopefully also use the models to evaluate if we are given certain photosynthetic traits, how would this crop perform in certain environments and such. And so by working with, by trying to think about ways to link photosynthesis with crop yield, we really have to focus on looking at the traits that are determining crop yield, and how do they interact with other processes in your plant. And recognizing key processes involved for the question at hand, of course, is important. And that for our case here, it would be the whole crop. And to get growth from photosynthesis, the crop that grows a canopy. If you grow your crop in a canopy configuration, you have to model the canopy, how it intercepts light, and how much light gets passed to your leaf. And I have a smiley face here because how many times do you get to work with legends in the field? And also biochemical models and enzyme equations are my favorites. So we have to think about the processes that's underlying grain yield, and also linking them, right? So we have to link photosynthesis at the leaf level to the canopy level, and this has to feed into this whole complex interactions at the crop level. And this is only half the story. The other half is how these complex interactions then feed back onto the canopy and changes your photosynthetic performance. And the modeling framework allows appropriate aggregations at the higher scales, and also let us zoom into the detailed mechanisms of photosynthesis. And I think we think this is what's needed to address the G/E/M interactions. And just to give you an example, you have a growth in your crop, gets more leaf area, but then your light interception per leaf area goes down. But because you're a bigger crop, so you do more photosynthesis and more transpiration, which dries up your water demand, but your water, soil, and nitrogen goes down, and then your stress comes up. And this would maybe just happen for one day, but what happens over a season? And I think it would be best to show you some simulation scenarios. And I say the 8 panels here. We're getting very good at looking at these 8 panels of trajectories of your crop traits. And what this allows us is to scrutinize the dynamics of G/E interactions for whatever issues that we are dealing with. And here, for example, for drought stress, I only have 4 panels here because I don't want you to go through the pain that I go through every day. 4 panels is enough here. So if we're looking at drought stress, so we're simulating a crop. Your crop would grow biomass over the season. Its LAI would go up, so leaf area per ground would go up and then come down. And if you look at your soil moisture, it comes down as your crop uses up the soil water. And what happens here, this is for a wheat crop, by the way. And what happens around flowering time is that when your soil water gets down to a level, it is not able to supply enough water for the plants to grow. So it hits stress. And when this happens, stomata closes, your biomass growth slows, and you hit a nosedive in your leaf area, which then further slows down your whole growth. And keep in mind that in the middle of all this, it is really the biochemistry, the photosynthesis, and then the canopy that's driving the biomass growth and the water demand. So here I'm showing you an example of how linking photosynthesis mechanisms would turn out if you, in a whole crop simulation. Now, of course, the individual responses are known. Water stress reduces your stomata and reduces your CO2 carbon gain. But the important thing is that integration is the normal thing here. It tells you when things happen and at what intensity, so that we can predict the outcome at the end. And so Kathleen talked about the functional plant biology paper that got the Best Paper Award in 2018. And that is the paper where we described a simulator or a model that gets us from the leaf level to the canopy level. And I've recently put this into an Excel, Microsoft Excel, and you can run it with Excel. It's available to anyone. And if you're interested or have any questions, you're happy to receive your email. But because I don't have much time, so I won't be going into the details about the canopy modeling. The thing I want to talk about is, okay, so now we have the cross-scale model linking leaf to crop. Is the model reliable? That's the big question. And to answer that, we compare predicted versus observed biomass and yield. And in this case, I'm showing here. And this includes close to 200 water by nitrogen treatment collected over decades of experiments. And so it just shows you the wide-ranging the yielding levels of these the experiments covered. And if you look at the predicted versus observed, it's pretty close. It's pretty close to the 1-to-1 ratio, indicating that the model can reliably predict different levels of biomass and yield for C3 wheat and C4 sorghum. And we have really scrutinized the 8 panels. So looking at the dynamic G by E over the season, and these experiments here, they're really high quality has high quality experimental data, which gives you this time evolution of your crop attributes, which we can then compare with the 8 panel analysis. So the conclusion here is that yes, the cross-scale model does work, and it reliably predicts crop biomass and yield for the right reasons. So having building the model and testing it with experimental data, and I think we're now in business for predicting crop photosynthesis for the synthetic manipulation outcomes, and hopefully learn some lessons from the predictions. So when we apply, so we can know when we apply some genetic changes at this level, we'll be able to tell you something about what happens at the grain, at the whole crop grain response. And for this, we've set up a simulation study where we looked at changing photosynthesis in sorghum crop, for example. And we've tested key putative photosynthetic manipulation targets by increasing the relevant photosynthesis models by 20%. So if we are looking at rubisco, we're looking at transport or the internal conductance or diffusion of CO2, if we assume that these processes were enhanced by 20%, And what that does to the ACI curve, photosynthesis versus CO2 response curve, is it pushes up the curve, so you get more photosynthesis for the same CO2. And so we've incorporated this into the cross-scale model simulation for a sorghum crop, and we ran the simulation for 120 years of 120 crop cycles at this location, and these are the standard sorghum sowing or growing conditions. And so what we found was that depending on the routes that we go about changing photosynthesis, the response to yield, so this is yield, averaged over 100, so change in yield averaged over 120 years. Depending on the routes that we go with, the response varies. But the biggest thing here is that as we go going from well-watered environment to the water-stressed environment, we're actually surprised to see that on the water-stressed environment, yield actually goes down a little. And we are puzzled about this this simulation results. So we looked into the 8-panel analysis again. So here I've singled out route number 2, where we change the enhanced electron transport. So the original simulation was highest for the well-watered environment, and in the average environment is somewhere in the middle, but in the water-stressed environment, your yield goes backward. And so what happened was during the water-stress environment, now with enhanced photosynthesis, your plants would be taking in more CO2, fixing more carbon dioxide. But what happens is it transpires a bit more, and this leads to the water stress around flowering time that I was talking about, and this effect has significant impact on the yield, which is driving the yield down for the water stress environment. So what we've stumbled across was from a note from Susanna was that, well, they observed that some transgenic plants with changed or manipulated photosynthesis, the stomata conduction didn't correlate with photosynthetic capacity. So what this is saying is you can have higher photosynthesis, but the transpiration rate might be the same. And so we've incorporated this into the simulation, and this rescued us in the lower water stress environment. And that's because by doing this, we are saving more water for the critical flowering grain filling period, and then we get the benefit of enhancing photosynthesis and not having to running into water stress, which decreases the yield. And we've tested this with other routes, so the 1 to 5, and this all seems to be happening with them. And what this what this highlights is it's critical to understand what's the connection between photosynthesis and conductance under circumstances when we enhance or when we manipulate photosynthesis, because it really changes our simulated results here. And what we also need to do is we need to test any or other water saving strategies to really help us widen the options for crop improvement. So in summary, again, unraveling the complex G by E by M interactions and the capacity to do trait evaluation can greatly accelerate crop improvement. And I hope I've shown that in my talk today, and especially for genetic engineering. So manipulating photosynthesis mechanisms, looking from our results, it is promising for improving crop yield. And really my holy grail I'm hoping to look at is if I can develop and test these models alongside these bioengineered plants, that would be a really exciting research to be doing. And I get to look at much more G by E by M interactions, much more of these to unravel. And again, we need to do more simulations and look at more eight panel analysis to really help us design better photosynthesis for crop improvement and moving beyond empiricism. And the question you might have is, can this be applied to other traits in other crops, in wider environments? The answer to all of that is yes. And we just have to advance the modeling to connect the processes across the scales, which is critical. And with this, I would like to finish my talk with thanking the collaborators who has been inputting in the development of this model. And thank you for your listening.
Thank you, Alex. That was a very interesting talk on a very complex issue, and you've really shown the power of mathematical modelling there. I have a question on the question and answer thing. Could you comment on the possibility of using genomic and other omic data in your prediction model?
Yes, that is actually something that our group is looking at. And I think the key to enable that is to find out, well, to develop models, maybe not in the whole cross-scale model batch, but a model that can help us predict, for example, Rubisco carboxylation rate or maximum electron transfer rate from genomics. Mm-hmm. And once we have that established, we can then use this model output into the cross-scale model to look at the G by E by M interactions for the crop scale.
Okay, do we have any, thanks Alex, do we have any other questions from the audience or attendees? Thanks, Roz. Roz is asking, Alex, do you think it will be possible to uncouple water loss from photosynthesis? It just seems very complicated.
Well, like I mentioned in the talk, so a paper that Susanna helped me notice was the transgenic study where they downregulated Rubisco, and in that paper they didn't see any change in stomata conductance. And so, I mean, I think this area needs to be researched a bit more to help us understand. So I would like to work with biochemists or molecular biologists to look at ways of uncoupling photosynthesis with stomata conductance, because as my simulation shows, that's actually quite critical if we want to grow our crops under these dryland conditions, which are mostly, well, yeah, mostly water stressed.
Yes, I can see that. Okay, thanks, Alex. From Peter Ryan, what was the transgene that disconnected conductance and photosynthesis to some extent in Suzanne's study? Oh, so, and I think you probably answered that, yeah, okay.
Yeah, it's the Rubisco downregulated tobacco plant.
Okay. All right, I think in consideration of the time, Alex, I think well, thank you for a very interesting presentation. You presented quite a complex issue in a very clear way. Thank you. So, good. Okay, so we'll move on to our last presentation for the afternoon by Karen Frick. Karen is the winner of the ASPS-FBP Functional Plant Biology Best Paper Award for the year 2020. So the Australian Society of Plant Science has a close association with the journal Functional Plant Biology, and every year they fund an award for the best paper from an early career researcher. So Karen was the award winner for 2020. I could tell you the title of her paper, but I'm sure that's going to come up in her talk. So Karen, could you please upload your presentation and come online? Thanks, Karen. And I would really like to thank everybody who has spoken today. It's been a fantastic set of talks. such excellent quality science that's going on here in Australia in plant science. It's fantastic and something that should be celebrated. And it's fantastic that we had this opportunity to have this session online for all our members and interested people to come and hear and experience that. So I'd like to end up by thanking you all again, thanking the attendees for attending and participating. And I would like to finish off by mentioning that the Society is having its AGM tomorrow. I'd like to encourage everyone to come along and attend. We have another award that wasn't mentioned today. That's the Teaching Award. There'll be a short presentation about that as part of our AGM tomorrow to make sure that's included and celebrated. That's on at 12 p.m. Eastern Standard Time. That's the 1st of October. And yeah, the last thing I want to mention was to thank Mary again and to thank the American Society for allowing this to carry on. And we've had such a great level of attendance, a great performance, and it really bodes well for the future if we have to continue on with this or even some sort of form of hybrid. So there's something going on in the chat here. I hope everybody, all the presenters are looking in the chat because there is a great response coming back to you and to this session. Yep, thanks everybody. And with that, I will draw this session to a close. And thank you all again.